today we'll be talking about genome editing. So, all living things, including ourselves, live under one genetic code, the DNA. The code that defines and differentiates between a human, plant, animal, and bacteria. The code that gives the genetic and physical traits that all living things live under till today, as we see here. Since the discovery of the DNA structure, the scientists have had the interest to try and locate the trait genes in order to modify them, insert new ones, or even delete them. And since then, genome editing has been born. They firstly tried to radiate plants and randomly mutate them in the hopes of finding good mutations. And then they tried to do double-strand breaks in many organisms to find changes. Then, when the restriction enzyme was discovered, scientists were able to remove DNA segments and replace them by others, which follow two similar techniques, the zinc finger nucleases and the TALENs. But these techniques were first used on model organisms, mice and monkeys, since they shared the same human genes, they shared similar human genes with humans. And then they tried it on model organisms like yeast, in order to knock out some of the genes and replace it by others. Sadly, these techniques were inefficient, expensive, they took so much time, and sometimes they were so difficult that scientists refused to use them in their labs. This was until 2002, when a study in bacterial immunity had discovered a loci called CRISPR, an adaptive immune system that specializes in keeping record of the viral DNA of viruses that attack the virus, that attacks the bacteria. So when the virus would attack again, the CRISPR would send a protein called Cas9 that would have the address of the recorded viral DNA and would seek out the viral DNA and cut it off like molecular scissors with precision and degrade it. By 2010, scientists had discovered that the CRISPR-Cas9 complex could be reprogrammed to recognize any DNA sequence and cut it. And they also found out that the CRISPR-Cas9 was much more convenient, was more efficient, was faster, cheaper, and much more precise. CRISPR-Cas9 was a revolution in the genome editing technology. By being able to make any changes in the DNA can do so much more than other techniques. This is using scissors. So first of all, the CRISPR-Cas9 can cut in any, can change any physical traits in the organism. So the first thing that they tried to do was using on these mice. These mice are all the same, but the difference between these mice and the others that these mice had one change using the CRISPR we removed the gene that was responsible for the black coat of rats. And so these mice are the control ones, and these mice were removed, had no black coat genes. One change in the gene made this different. Even when the genetic analysis was done on these rats, they were all the same except one gene that was responsible for the fur. So what are the potentials? The University of Philadelphia was successful in eliminating, using the Cas9 protein, they were able to remove the HIV-integrated genes that were infecting the cells, using the Cas9 like scissors. And they were able to induce the DNA repair enzymes in regions that had cystic fibrosis mutation. So when we identify the region that had the mutation by cystic fibrosis, we can give the Cas9 sequence and do the double-strand cut. So the DNA repair enzymes would be induced to repair the region, and so there's no cystic fibrosis. So the first potential is disease immunity. Not only that, what if we can change the plants to not only grow in tropical areas, but in the Sub-Saharan deserts? What if we can make cows and other animals to give us yields to increase the yield more than average? Let's take it much more further. CRISPR can work on any organism, and not only adult cells, but embryonic cells. So we can change the embryos of our humans. We can change the babies themselves. What if we can change the physical traits of babies and give them blue eyes, blonde hair, or we can change the genetic ones and give them super strengths, or make them the next Einstein? This is all possible by CRISPR. And what if we can tamper with the genes of age and make people eternally young? They don't never grow. But this potential has raised so many questions. The ethical dilemmas, the people that are of the ethics refuse to let people try and tamper the babies. Social inequalities, if we can make designer babies, there will be social inequality between normal people and superior people. Bad intention if scientists use it in a bad way, and off-target mutation, simply if I'm trying to make Superman and he becomes Milky Man.